Hello everyone, welcome to E-Classes by Dr.Athira. Today I am going to discuss the structure of salivary glands and their ductal system. Salivary glands are exocrine glands that produce and secrete saliva into our oral cavity. There are three pairs of major salivary glands and 600 to 1000 minor salivary glands that secrete saliva into oral cavity through their ductal system. So now let me move on to the topic of discussion, that is structure of salivary glands. Salivary gland consists of epithelial components as well as supporting connective tissue. The epithelial components include acini or terminal secretory unit, their ductal system, and a specialized epithelial cell known as myoepithelial cells, which are seen associated with the acini. An acini is basic functional unit of salivary gland and its terminal secretory unit. There are two basic types of acini. One is serous acini, other one is mucous acini. The secretions of both differ. Secretions of serous acini are more enzymatic and less viscous in nature, whereas mucous acini or mucous cells produce viscous carbohydrate-rich secretions. So an acinus consists of few secretory epithelial cells along with the central lumen. The secretions from the epithelial cells, secretory cells, are secreted into the lumen and then carried to the ductal system of salivary gland. The ductal system of salivary gland is quite complex and all these ducts are lined by again epithelial cells. So these epithelial lined ducts carry the saliva into oral cavity. And there is another specialized epithelial cell known as myoepithelial cell seen associated with the acini as well as the terminal secretory duct. And all the structure, epithelial component of salivary gland is supported by surrounding connective tissue. Now let's see the structure of an individual serous and individual mucous acinus. As you can see here, the serous acinus and mucous acinus. The acinar cells or secretory epithelial cells are arranged in a spherical form in serous acini, whereas in mucous acini they are arranged in a tubular or ovoid form. Adjacent cells of acini are connected to each other by means of intercellular junctions. Intercellular junctions are present in both serous and mucous acini. The intercellular junctions, as you can see here, they help to hold the cells together in the acinus and also control the permeability of the acini. And the secretions from the cells is secreted into the central lumen. The central lumen is smaller in serous acini and is larger in a mucous acini. And if you see the shape of central lumen, it is star shaped in serous acinus because the lumen extends between the cells like this. These extensions of lumen in between the cells are known as intercellular canaliculi. The intercellular canaliculi is absent in case of mucous acini. And you can see here the myoepithelial cells present on the surface of acini, present in both mucous and serous acini. And there is a basement membrane covering the serous acini as well as mucous acini. The basement membrane separates the acini from the surrounding supporting connective tissue. You see here the myoepithelial cells are also enclosed within the same basement membrane. When we see individual serous acinar cell, the cells are pyramidal in shape with a basally located nucleus. You can see the apical cytoplasm is filled with lot of granules. These granules contain the synthesized components of saliva or the secretions that will be secreted into the lumen. So these storage granules are known as zymogen granules. But in case of your mucous acini, you can see the apical cytoplasm filled with mucus droplets. They are not stored in any granules, but instead the apical cytoplasm is totally filled with the secretion. This secretion will tend to push the nucleus towards the base of the cell. That's why in a mucous acinar cell, the nuclei is compressed or flattened in the basal cytoplasm. So in a purely serous salivary gland like the major salivary gland parotid and the minor salivary gland von Ebner salivary gland, we will find only serous acinar. But in case of a purely mucous salivary gland, there will be only mucous acini present. But in case of a mixed salivary gland, there will be both serous and mucous acini present within the same gland. There is one more structure to be discussed in mixed acini. For example, like in your submandibular gland, which is a mixed salivary gland, both the acini will be present and sometimes the serous acini may be found on top of the mucous acini, or mucous acini may be capped by the serous acini like this. This specialized structure is known as serous gangliones. Now let's see electron microscopic view of a serous acinar cell as well as a mucous cell. The serous cell, as you can see here, it's pyramidal in shape with its apex located towards the lumen of the acini. And the nucleus is situated in the basal end of the cytoplasm, and the apical cytoplasm contain lot of secretory granules of 1 micrometer diameter. These granules are known as zymogen granules, and they store the secretions or salivary secretions. And the cell, serous cell, has all the features of a cell specialized for synthesis, storage and secretion of proteins. So the proteins in the sizing apparatus include the rough endoplasmic reticulum arranged in parallel stacks basally and lateral to the nucleus, and prominent Golgi complex, and also there are a lot of mitochondria arranged basal and lateral to the nucleus. As I said, the cell is specialized for the synthesis of protein because the serous secretion is rich in enzymes and proteins and it's watery. And there are junctional complexes connecting the cells with each other. The mucous cells are specialized for the production, storage and secretion of carbohydrate-rich material, and the apical end of the mucous cell shows the secretion in the form of mucus droplets, and this fills the cytoplasm in the apical region, and also in fact this secretion, presence of this rich mucus secretion in the cell, pushes the rest of the cell organelles and nucleus towards the base of the cytoplasm. That's why the nucleus of a mucous cell is seen in the basal end and it's seen flattened or compressed. And the mucous cell is containing prominent Golgi complexes as it reflects cells' increased carbohydrate metabolism. Other organelles are less prominent when compared to serous cell because the cell is not involved in protein synthesis. And there are intercellular junctions connecting adjacent mucous cells, but these interdigitations are less when compared to or fewer when compared to that of serous cell. So in this acini stained section of salivary gland, here you can see mucous acini as well as serous acini. This is a acini stained section of a mixed salivary gland where both type of acini are present. There is obvious difference in the staining of serous acini and mucous acini. Serous acini are stained intensely with the acini stain as they contain secretory granules rich in proteins and prominent proteins in the sizing apparatus. So they stain intensely with hematoxylin stain, whereas the mucous acini you see it's pale because the carbohydrate rich cytoplasm do not take up the hematoxylin stain. That's why the mucous acinar cells or mucous acini all appear pale. Other than serous and mucous epithelial cells, there are a specialized group of epithelial cells known as myoepithelial cells in salivary gland. The myoepithelial cells are present associated with the acini as well as the first duct starting from acini known as intercalated duct. And you can see these cells are enclosed within the same basement membrane of acini and the duct. These myoepithelial cells resembles an octopus sitting on the rock, as you can see here. It is located on the surface of the acini and they have a long processes, thus has a star or stellate shape. But on the surface of the intercalated duct, you can see they have short processes, thus has a spindle shape. Now, what is the role of these cells? Myo means muscles. So these cells contain actin and myosin intermediate filaments which has contractile property. That's why they are named so. The contraction of these cells causes the passage of saliva secreted from the acini outwards towards the duct and towards the oral cavity. And also they support the acini and the duct and prevent their collapse, create a positive pressure and prevent the backflow of saliva. So the acini of salivary gland, which are arranged in the form of lobes and lobules along with myoepithelial cells and also the ductal system, makes up the epithelial component of salivary gland. The supporting connective tissue surrounds the entire salivary gland. Also, they run in between the lobes and lobules of acini as connective tissue septa. So, thus the entire structure of salivary gland can be simulated to a bunch of grapes, and the grapes represent the acini and their stems represent the extensive ductal system of salivary gland. The larger duct opens into the oral cavity and towards the secretory NPs, they divide into various interlobar and interglobular branches connecting every single acinus. The ductal system of salivary glands are not just a mere passageway of saliva, but also they take part in active modification of secretion. They consist of hollow tubes connected initially with the acinus, as you can see here, and then progressively grow larger from inner portion that is near the acini towards the opening into oral cavity. Each major salivary gland displays difference in length of different types of ducts. There are 3 different ducts differing in its diameter, type of epithelial lining and in its function of modification of saliva. The first duct starting from acini is known as intercalated duct, which is continuous with other 2 ducts is that the striated duct and then the excretory duct which opens into oral cavity. So the intercalated duct starting from the acini and then the striated duct and excretory duct forms the ductal system of salivary gland. Acini of salivary gland is arranged in lobes and lobules. Each lobule is surrounded by supporting connective tissue. These 3 ducts can be named even according to its location as intralobular and interglobular ducts. Intercalated duct and striated duct, which are located within the lobules, are known as intralobular ducts, and the excretory duct, which is situated in between the lobules, are known as interlobular duct. As I said earlier, the ducts are lined by epithelial cells, but there is difference in diameter and nature of epithelium lining these 3 different ducts. So now let's move on to it. The intercalated duct, which is the first duct starting from the acini, you can see here. It is the smallest diameter duct. They are lined by small cuboidal cells. You can see here the duct lined by small cuboidal cells. There is presence of myoepithelial cells associated with the intercalated duct, or they are present on the surface of the intercalated duct. You can see here. This was explained earlier also. You can see in this acini section of salivary gland presence of intercalated duct, which is the smaller diameter or smallest diameter duct among the salivary gland ducts. When you see individual cuboidal cell lining the duct, you can see the nucleus located in the center. There are occasional secretory granules also present in the intercalated duct. That means they also take part in the secretion of saliva to an extent. Intercalated ducts are prominent in salivary glands having watery secretion, that is serous secretion. For example, in parotid gland, intercalated ducts will be more prominent. The intercalated duct passes their secretions into striated duct. You can see here the striated duct are larger in diameter when compared to the intercalated duct. Intercalated duct is lined by the cuboidal cells, but the striated ducts are lined by tall columnar cells. And another very important peculiarity of striated duct cell is it has lot of indentations or infoldings of the basal cell membrane. You can see the cross section of striated duct in this acini stained section. The cell has a centrally placed nucleus and eosinophilic cytoplasm, and the basal cell membrane shows lot of infoldings, and there are presence of mitochondria in between these basal infoldings. Altogether, this gives a appearance of striations, prominent striations, thus the duct derives its name, striated duct. There are few apical secretory granules present, and below the cell, the connective tissue, there is presence of rich capillary plexus. This rich capillary plexus below and this regular striations give it a name as pin necklace. The striated duct passes saliva to the larger excretory duct. Excretory duct are the largest ducts. You can see they have larger diameter, and they are the ducts that opens into oral cavity to secrete saliva into oral cavity. Initially, near the striated duct, the excretory duct lining are similar to striated duct lining, that is, they are tall columnar cells. But as they progress towards the oral cavity, they become stratified columnar, and towards the opening in the oral mucosa, they become just similar to oral mucosal epithelial lining, that is stratified squamous epithelium. Other than the ductal epithelium, there is goblet cells, brush cells, and ciliated cells also occasionally in the excretory duct. And the connective tissue surrounding the excretory duct might show abundance of elastic and collagen fibers, which allows passive stretching of ducts to accommodate varying volumes of saliva. In this cross section of duct, you can see acini diagram shows multi-layered or stratified squamous epithelial lining of striated duct. So that's all about the structure of ducts, and coming to the functions of ducts, I told already they are not just conduit or passageway of saliva, but they also are involved in modification of saliva. The saliva secreted from the acini gets modified in various ductal system. So let's see how is that happening. The intercalated duct contributes macromolecules like lactoferrin and lysozyme to saliva from ductal cells. These are antimicrobial substances in saliva. The water content of saliva enters almost completely at this level of intercalated duct. So the intercalated duct cells contribute lactoferrin, lysozyme into saliva, and also almost all of its water content. The saliva secreted from the acini and the intercalated ducts are usually isotonic or slightly hypertonic in nature. And another small point regarding intercalated duct is that it also is thought to house few undifferentiated epithelial cells which can replace the damaged salivary gland epithelial cells. Saliva from the intercalated duct passes on to striated duct. As we have discussed already, striated ductal cells are highly characteristic structurally. The cells lining the striated duct, the tall columnar cells, have got lot of basal cell membrane infoldings which increases their surface area, and there were a lot of mitochondria present between these infoldings within the cell. Mitochondria provides energy for active transport of material, and there is greater amount of reabsorption and secretion of materials in saliva at the level of striated duct. There occurs reabsorption of sodium and chloride from saliva and secretion of bicarbonate and potassium into saliva by the striated ductal cell. This process makes the saliva, which was initially isotonic or slightly hypertonic, into a hypotonic solution. The final saliva secreted into oral cavity is thus hypotonic in nature. Other than this, striated duct also synthesize and secrete organic components like kallikrein and epidermal growth factor into saliva, and they are also involved in small amount of protein reabsorption. Striated duct is relatively impermeable to water. So that's all about the important function of modification of saliva by ductal system of salivary gland. That's the end of this session. Thank you for listening.